Okay. Jump back in now for exam 3 material. Talking about oxidative phosphorylation. This was obviously more recent material, so hopefully it is all still very fresh in your minds. So, first of all, know what oxidative phosphorylation is and its purpose. So, oxidative phosphorylation is the process in which ATP is going to be formed as a result of transferring the electrons from NADH, FADH2 to an oxygen through a series of electron carriers. So this is going to take place in the mitochondria, specifically in the inner membranes or the cristae of the mitochondria. The purpose of this is to generate the majority of ATP in aerobic organisms to meet our energy needs. So talking about the enzymatic steps in the citric acid cycle produce reducing agents. And those reducing agents hold onto and pass electrons. So this is just a repeat, kind of giving us which steps give us these NADH and the FADH. Remember, I am the most important isocitrate, alpha ketoglutarate. I am, and then most important. Then just remember that F goes with F, so succinate to fumarate is your FADH2. Yeah, so we end up with those electrons. So those electrons are attached to our NADH and our FADH2, and those are high energy electrons that we're going to be using to produce our ATP. Before we get into that, kind of how we actually produce the ATP, we need to talk about these different shuttle systems. Because once we produce the NADH, we need to have a way, specifically with glycolysis, that NADH that we produce during glycolysis, we need to get it across the membranes to get it to where it needs to be. And we have to get it all the way from the cytosol and get it through the membranes and into the matrix, in the mitochondrial matrix, in order for it to then do its job in the electron transport chain. So these cytoplasmic shuttle systems are going to transport the NADH across the inner mitochondrial membrane for the oxidation. Now there's two different shuttles. There's the malate aspartate shuttle and then the glycerophosphate shuttle. So the malate aspartate shuttle, we have the cytosolic NADH, reduces oxaloacetate to malate, and then it's going to be able to cross over the membrane in exchange for an alpha ketoglutarate, thanks to the malate alpha ketoglutarate carrier protein. Once it's there, it's going to reoxidize and give the electrons across. And so now we have NADH inside of the membrane, and then the oxaloacetate goes back and actually becomes aspartate, so that it can cross back over, and then it repeats in a circle over and over and over again. There's the step-by-step in words, if you want to pause and read that, just so it's a fairly straightforward process. Looks a little more complicated just because of the long names and stuff, but it's really not that bad. Next, the glycerophosphate shuttle. Let's see if I can get this all to fit on one page. There we go. So the glycerophosphate shuttle is cytosolic dihydroxyacetone phosphate is going to be reduced to 3-phosphoglycerate and forms NAD+. The 3-phosphoglycerate is oxidized by an intermitochondrial enzyme and delivers electrons directly into the electron transport pathway. So we have our NADH. We use a glycerol 3-phosphate dehydrogenase, and that passes it on to a glycerol 3-phosphate, which then we have that flavoprotein dehydrogenase, which is going to transfer those electrons to an FAD, turning it into an FADH2, and that is going directly into the electron transport chain. If I remember correctly, the glycerophosphate shuttle specifically happens, I want to say in the brain. Let me double check. Glycerophosphate shuttle occur. That was it. The glycerophosphate shuttle is used in the skeletal muscle and the brain, and the malate aspartate shuttle was in the kidneys, the lungs, and the liver, if I remember correctly. Big one to remember: the kidneys and the liver. This happens in the liver. Now the electrons being transported through the electron transport chain and how they're transferred to O2. Here is our here's a much better drawing than what I had before. So the whole process, we have our different complexes 1, 2, 3, and 4. A couple different cytochromes to help us out. CoQ, also called ubiquinon. We have our NADHs. And so the process of this, the NADH, is going to give its electrons to complex 1. FADH2 gives its electrons to complex 2. And both of those are going to be giving their electrons to this CoQ, this ubiquinon. Complex 1, at the same time, when it's holding on to those electrons, it's actually going to use the energy that it receives from those electrons to pump hydrogens through from the matrix into the intermembranous space. And then it'll pass them off to the CoQ. But it's complex 2 also passes it off to the CoQ, and it travels to complex 3. Complex 3 goes into cytochrome B and then cytochrome C1. And then eventually pass them off to cytochrome C, which then continues and carries it to complex 4. When it's here in complex 3, it does the same thing. It pumps hydrogens through. And complex 4, it gets past to cytochrome A and then cytochrome A3. And when that happens, it also pumps a couple more hydrogens. So now then finally, once it is done, it gets to the end of the line here. And the electrons are going to be passed off to an oxygen in order to form H2O. Now, the whole purpose behind passing this electron down and pumping the hydrogens was to create an electrochemical gradient. Basically, just putting a ton of hydrogens up here at the top, so there's a much higher concentration up here than it is down here. So then, this ATP synthetase, or complex 5, has an opening, and it's going to when there's a whole bunch of hydrogens up here, those hydrogens, because they want to go from high concentration to low concentration, we're going to use this as a channel. So the hydrogen are going to flow through here, and inside of this this protein, there is kind of like, you can think of it like a turbine or like a fan blade almost, and it's going to cause it to spin. And as it spins, it converts the electrochemical energy into movement energy, and then that movement energy is used to take ADP and inorganic phosphates and combine them to create the bond to form ATP. And that's all the electron transport chain. It seems like a lot of steps, but once you go through it a couple of times, it's not as bad as you think. Continuing through, we have knowing the components of the electron transport chain. Again, we just discussed all of those: complex 1, complex 2, 3, and 4, the different cytochromes, the coenzyme Q, and then the ATP synthetase, aka complex 5. Let's stick with Roman numerals. So this right here, we can see all of our, let's leave it down there. Kind of all of the, little bit more the fancy kind of organization the way it was in the slides. Personally, I don't think that this is as simple. I think that this, just looking at it like this, is much more simple. And just understand the oil rig. Oxidation is loss of electrons, reduction is gain of electrons. So you're oxidizing these different things. So it has an oxidized state and a reduced state. As it passes the electrons along, it gets, like the complex 1, when it passes it to CoQ, CoQ gets reduced and complex 1 gets oxidized, and then CoQ gets oxidized when it passes it off to complex 3, and complex 3 gets reduced, and so on and so forth. There's the flow of electrons, how the electron transport chain generates protons. So that proton gradient and the protons themselves, the protons are coming from the NADH and the FADH2, and that's occurring from glycolysis and from the Krebs cycle. However, so the gradient itself, we talked about that, how the different, specifically complex 1, 3, and 4 pump the hydrogens across using the energy provided by when they are reduced with the electrons. And then this is right here. The exergonic return of the hydrogen to the matrix powers the ATP synthesis. The hydrogen passes through ATP synthetase and drives turbines. This movement gives the energy needed to convert ADP to ATP. There is more to this. Like there's technically like alpha and beta parts of the ATP synthase, and there's like a rod and a drive chain, like a driver, like a, not a drive chain, but it's like there's like a rod, and the rod spins, which then causes something else to turn, which causes other stuff. It's going a little bit deeper though. If you're interested in this, Ninja Nerd has a really great video on electron transport chain. I really enjoyed watching it, going through it. It dives a little bit deeper into the material than we need to go. So, yeah. What is the chemiosmotic hypothesis? Is literally what we just talked about. It's describing the coupling of the electrochemical gradient to ATP synthesis. The whole process that we just talked about is the chemiosmotic hypothesis. ATPase is using the free energy electrochemical hydrogen gradient to form ATP. The enzyme responsible for ATP formation during oxidative phosphorylation is the ATP synthetase, aka there's some others that you'll see, ATPase or oxidative phosphorylation. How many moles of ATP are formed from 1 mole of NADH or FADH2? We've been over this a whole bunch of times. 1 mole of NADH is 2.5. 1 mole of FADH2 is 1.5. Coupling of the electron transport chain, reduction of O2 to H2O, and ATP synthesis in the oxidative phosphorylation. Again, these questions are all saying the exact same thing, just in different ways. Where the electrons pass through, you get the electrochemical gradient, and then once it goes through complex 4, through those cytochrome A1 and A3, not A1, sorry, just cytochrome A to cytochrome A3, and then out. There needs to be some place for those electrons to go now that we've used up a bunch of their high energy and stuff. And it's the easiest thing is we produce water. Examples of uncouplers of the oxidative phosphorylation process. Apologies, give me one moment. The wife is working tonight, so I'm the only one that can give attention and treats. Okay. The uncouplers of the oxidative phosphorylation, essentially the things that mess with the oxidative phosphorylation and prevent it from happening. We have DNP and FCCP. Let's goes over how. There wasn't a ton of questions about uncouplers. I wouldn't concern myself a ton with these. But the uncouplers block the oxidative phosphorylation of the ADP to prevent ATP synthesis. So all of the other steps before this happen, but instead of it turning into ATP, the chemical energy is converted into heat energy to keep the individual warm. The one that you do want to know is right here. This thermogenin is a natural uncoupler present in brown fat. It says of many hibernating animals, also of infants. Babies have brown fat. Essentially, what it's doing is the entire electron transport chain is going to be running, doing its thing in those mitochondria. But rather than producing ATP and energy, it's just producing heat. It's to maintain the body temperature of the animals without having to produce a whole bunch of energy. So if we think also like hibernating animals, the hibernating animal doesn't have any way to use that ATP, and if they had a massive buildup of ATP, then it would shut off and it would stop producing. So we need those that uncoupling, that stopping of just the production of ATP to continue to produce heat in order to keep the body temperature up during hibernation. We also have some specific blockers. Rotonin, antimycin A, cyanide, and carbon monoxide are all blockers of the electron transport chain. This again was in your exam. If you get questions on this, the questions are going to be the exact same as the ones that you had on this exam. So I think there was a question about antimycin, and I think there was also one about cyanide, if I remember correctly. Might be wrong on that, but yeah. Lecture 12. The pathways of sugar metabolism. The metabolism of fructose, galactose, mannose, and the pentose phosphate pathway. So, a whole bunch of this. I'm going to be honest. A lot of this I didn't feel was super drilled heavily. However, I would recommend you go back to let me check which chapter specifically is in the book. It's chapter 12. So the Biochem 1, chapter 12, metabolism of sugars. It's only 13 minutes. If you want to go, I would go back, review that, go through what I would read in the book, because that's exactly what's going to be here. Just go over the sugars and how they go through, or not. I mean, he might throw some stuff on here about this, but given all of the different information, everything that you have, I don't see it being as tested as some other topics. Now, obviously, take that with a grain of salt. It's been a while since I've taken the class, but there's much better testable material with a lot more questions stemming from them than this specific material. Maybe remember the the monosaccharides. If we think about the glucose, trying to remember the names of the sugars that we talked about. I think it was there's maltose, lactose, and sucrose, and kind of what each of those are split up to. So we have sucrose, lactose, maltose. Yeah, those are the main ones that you'll see, and what each of those are made up of. Mannose metabolism. Okay, pentose phosphate pathway. This is the more important stuff. So, importance of the pentose phosphate pathway is to maintain the carbon homeostasis. It provides precursors for the nucleotide and amino acid biosynthesis, so for DNA and protein biosynthesis. It provides reducing molecules for anabolism and defeat oxidative stress. So it's responsible for NADPH production for the synthetic purposes, for biosynthesis. The NADPH is used for reductive biosynthesis to create fatty acids, sterols, those cholesterol type things. We'll learn more about those at the very beginning of Biochem 2, and also the synthesis of macromolecules. Formation of ribose 5-phosphate, that is required for nucleic acid biosynthesis. So how does it produce the NADPH? Let's go find the wonderful picture. Put in all of the extras and stuff there. Good. Okay, so the oxidative phase. Oh, there's two different phases. There's the oxidative phase and non-oxidative phase. So we have glucose 6-phosphate. We have our different enzymes that are going to act on, and very similar to just like in glycolysis, the dehydrogenase enzymes are going to take the NADP and form it into NADPH. Same thing happens here. Then we're ribulose 5-phosphate. If we think back, ribulose versus ribose. Remember, ribose was the name of the sugar. Ribulose is the ketone, and the ribose is the aldehyde, which we talked about before. And then through here, the ribose 5-phosphate, we can go and have either trans-ketolase or the trans-aldolase. We'll convert it back to glyceraldehyde 3-phosphate and fructose 6-phosphate, which can then go back to glycolysis if needed, or it can continue and be turned into the nucleotides, coenzymes, and DNA and RNA. So it just depends whether it's needed or not. Yeah, and that's the pentose phosphate pathway. Lecture 13, how much are we doing on time? How long is this chunk? It's not too bad. We'll see if we can finish this first before ending this section. Okay, first of all, what is glycogen? Glycogen is the storage form of glucose in animals. What was the storage form of glucose in plants? Starches. So we have the glycogen is polymer of glucose, meaning that it's a whole bunch of glucoses stuck together. There's two different types of bonding. There is a linear chain, or linear bonding, and a branch chain, or a branch bonding. The linear chain, the 1 carbon binds to the 4 carbon of the two different glucose molecules, whereas in the branch point, the alpha 1 carbon, or the 1 carbon binds to the 6 carbon on the glucose. And the reason why it forms these branch changes is that you can fit a lot more glucose in this way, or a lot more glycogen in this type of bonding, than if you were to just do one long linear chain. So it takes up less space. So how does muscle use glycogen stores and glucose? So the need for ATP triggers the conversion of the glucose to G6P. Muscles don't have super big stores of glycogen, so like 1 to 2 hour intense workout, you will use all of it. The muscle uses all glycogen in its own cell. It does not release it. The liver will, in fact, release it into the blood, though. So the liver is in charge of maintaining our blood glucose. Glycogenolysis. Do we have glycogenesis here as well? Let's talk about glycogenesis first. So the process of glycogenesis, our hexokinase in the muscle or the glucokinase in the liver, are going to convert the free-floating glucose that's in the blood to G6-phosphate. It's going to phosphorylate it as it brings it into the cell. Phosphoglucomutase converts it to G1P, which is the storage form of glucose, or it's the form in which it takes in when it so that it can be stored. There's the next enzyme, UDP glucose pyrophosphorylase, converts G1P to UDP glucose. Glycogen synthase is then going to catalyze the transfer of the glycosyl residue of UDP glucose to a non-reducing end of a branched glycogen molecule. And that makes a new alpha-1,4 linkage. There's another enzyme called the glycogen branching enzyme, which makes that 1,6 linkage. And we can see this happening here. It's going to have a non-reducing end and a reducing end. Is technically over here. We're going to continue to build off of the non-reducing end. So you can see that 1 to 6 branch point. That's done by the branching enzyme. And this is how glycogen gets built. Make sure. So the glucose residues are added to the non-reducing site of the growing glycogen chain. Glycogenin is what is going to prime the initial sugar residues in glycogen synthesis. So for glycogen synthase to start, a pre-formed alpha-1,4 polyglucose chain having at least 8 glucose residues is needed. The glycogenin makes that initial structure so that glycogen synthase can activate. So the glycogenin is going to put together 8 glucose residues, put together 8 glucose to kickstart the process. Regulating it due to covalent modification, so phosphorylation, glycogen synthase gets inactive. Dephosphorylation, glycogen synthase is active. Stuff like insulin, presence of G6P and glucose are all activators. The inhibitors are the glucagon and epinephrine. Glycogen synthase kinase 3 is going to stop glycogenesis. So the kinase does phosphorylation. Phosphorylation inactivates it. And then our phosphoprotein phosphatase is going to remove that phosphorylation in order to activate that enzyme. Okay. Shifting back to glycogenolysis, about breaking it down. Our different enzymes, we have glycogen phosphorylase. This is going to break down the glycogen in the cells. It's going to break off glucose from the non-reducing end of the glycogen. So it travels along and it just like snips them off, like snip, 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 one at a time from that non-reducing end. The debranching enzyme, so when there are only 4 glucose left on a branch, it takes 3 and it's going to move it to the next branch next to it. And so the last glucose that forms the branch point is hydrolyzed at the alpha-1,6 linkage. So then that last glucose, so it takes 3 of them, and then it breaks off the last glucose that's still attached, officially removing the branch, that specific branch of glycogen. So phosphoglucomutase, then it's going to convert the G1P back to G6P in both the liver and the muscle in order for it to then go do its job of either producing ATP or getting released into the bloodstream to maintain our blood glucose. If we already talked about the phosphorylated enzyme, glucose 6-phosphatase. This is after the phosphoglucomutase. It's converted from G1P back to G6P. The glucose 6-phosphatase needs to break off that last phosphate so that it can go and be let back into the bloodstream. So this is going to happen in the liver. The glucose 6-phosphatase removes that phosphate so that it can go and travel through the cell wall and go back into the blood and maintain the blood sugar of the body. The regulation of glycogenolysis, glycogen phosphorylase. Again, you can think a lot of these are very, very similar. Just stop and think what's actually happening. If we're releasing a bunch of glucose into the blood, it's because we need more energy. If we need more energy, what kind of stuff are we deficient in? We're deficient in glucose, in the glucose 6-phosphate. We're deficient in ATP. We'd be deficient in NAD and FAD, etc., etc., etc. And then again, the reverse is true. Like if we have a bunch of energy, we have a bunch of extra glucose or a bunch of extra energy, we have absolutely no point to be forming glycogen, and so it'll be going in the opposite direction. It'll be being inactivated. I feel like this part right here was not in the book when I remember being there. So this is the phosphorylation and dephosphorylation in comparing the myocyte versus the hepatocyte. This might actually be a picture that I found outside. But yeah, this goes through the kind of the chain reaction that occurs to activate it. You can see you can see the cascade effect happening as we go down. We talked about signaling just a little bit ago. That's what that's referring to, how the signaling just exponentially grows. Okay, we talked about glycogenesis. Hormonal regulation. Epinephrine versus glucagon. Epinephrine in the muscle. Epinephrine is adrenaline. So if we have a lot of adrenaline, it's our fight or flight. We need a lot of energy, so it's going to break down glycogen. Glucagon stimulates the glycogen breakdown and gluconeogenesis and blocks glycolysis. Some of our glycogen storage diseases: von Gerk's disease, Pompe's disease, Forbes or Corey's disease, Anderson's disease, and McArdle's disease. You can just kind of lump these and see. Just remember that what enzyme is affected by these. Okay. We'll go ahead and do another pause here. We are getting very close to the end. Let's see kind of what else we have. Ethanol metabolism, nucleic acids in DNA. All that DNA stuff and the RNA structure and transcription. And the very, very end should be translation. Yeah, all that in translation. There's not a whole lot left. Hopefully we can finish wrapping this up in one last part. That'll keep it around 2.5-ish, 3 hours or so for total, which hopefully isn't too much. Okay, see you in the next one.